Okay, just want to welcome everyone here. This workshop is Silkworms as an industrial organism from Martian Biosystems.
All right, thank you. And hello, everyone. I'm Stellie Ford. So I'll be giving the presentation tonight. And give me just a moment. I'm going to share my screen so I can share my presentation with you. All right. And feel free to raise your hand via the Zoom system at any point in time if you have any questions, and I'm going to leave a bit of time at the end here too. Okay, so as mentioned, the topic tonight is silkworms. And so we're looking at them from the broad context as an organism that can help produce a lot of goods and materials that will be very useful on Mars. And I'm here to tell you exactly how that can be done. So first, okay, so first for a bit of background on me. So I've studied bioengineering and tissue engineering a bit more specifically, and I'm working in biotechnology, biomedicine at the moment, specifically more the technical side on water systems and dialysis technology. So I've been very active in the Mars scene over the past couple of years since getting involved via the colony contest in 2019. I'm leading the Philadelphia chapter of the Mars Society at the moment, which is one of the more humble chapters, but we're all very passionate about the biomaterials, the biotechnology, the life system side of colonizing Mars. And so I think you'll get a lot of that in tonight's presentation. And so for those of you who might be a bit more visually oriented, this is just an overview of what I'm going to be covering today and a bit of background for anyone who might want to look into that a bit further. Okay, so first I want to give you guys a bit of background on silkworms. So they're a very easy organism to grow. You can actually get the eggs online, and that's really the main way to get them unless you're growing them yourself, in that there are no silkworm farms in the US anymore, so you have to get them shipped to you. But that's okay. So typically in the wild, silkworm eggs can go through a diapause phase where they'll hibernate over the winter. And then this actually allows for easy cold storage and shipment because of that. But if you're raising them yourself, you can continue to propagate them without the cold phase, or you can manually induce the cold phase by putting them in the refrigerator. But overall, they have about a 2-month life cycle, and they're very easy to grow in captivity. And so they just need to be kept warm and given mulberry powder mixed into a chow or fresh mulberry leaves as their food. But as I said, the whole process takes about 2 months. So there is not a great survival rate, particularly in the early hatching stage. You're looking at roughly 85% of your eggs to hatch, but you'll probably only see about 50% of those worms make it to full maturity in the bin, especially in the earlier stages. Industrial farms have a much higher survival rate and production rate, but just a bit of background for anyone interested in farming silkworms at home, like I've gotten into. So when you get to the slightly larger industrial scale, though, things really tend to step up, and particularly by combining different batches of silkworms from those different instar stages, you're able to then grow them in a much higher density. Really, all they need is for a fresh supply of food and chow every day to be happy and to be kept warm. And it's not too warm, it's in the 70s to low 80s, so it's very achievable with home heaters. Right. But you can get to a density of about 800 silkworms per meter squared, which is quite dense, particularly considering the fact that you're able to stack the trays that the silkworms are raised in, as you see on the right here. So this is at the cocooning stage. So these could be silkworms that are essentially past their larval stages, their instar stages, and have now reached the full cocooning, or this could just be a collection, but this is representative of the growing trays that silkworms live on and then can form their cocoons on before harvest. And so particularly when you're looking at facility level growth setup, where you're able to combine different instar batches, you can turn over a batch within 2 months pretty easily. So you'll be able to get 6 batches per year on each of these towers or tray sets, leading to a pretty dense growth rate for silkworms, about 128,000 per cubic meter per year, given average spacing. But notably, you do need quite a few silkworms to produce most goods. So it's that number is quite high, but not quite as high as it seems once you start getting into some of the numbers on that. As I mentioned before, the primary food for silkworms is mulberry powder or mulberry leaves. Other vegetables and fresh greens can be substituted in, but for healthy silkworms and for happy silkworms, you really want to use mulberry. But this is actually very easy to grow and incredibly easy to transport in a dry powder storage. And this is one of the biggest benefits for silkworms as an organism, is that they're very easily transported to Mars, and their food as well can be transported in bulk quite easily. A cubic meter of silkworm chow can raise tens of thousands of silkworms. And it offers a way to have a very large stockpile of their food without necessarily needing to commit to growing the mulberry leaves fresh, though that is quite possible. So there's minimal overall labor involved with the silkworms. It's essentially just feeding them and cleaning them, but particularly with the use of mats that allow them to crawl up onto a new layer of food, you can then transport them to a new tray and easily clean the old one without having to deal with the silkworms themselves. And the whole process is minimally time involved. So, but to move over to mulberry trees for just a moment, coppicing is really one of the best ways to focus on just growing the fresh shoots and leaves of mulberry, which are the things that the silkworms are going to be eating. So by focusing on coppiced farms, we're able to greatly increase the density of trees that can be planted in a given space. And so it would take about 2 hectares to support something like 2.5 million silkworms being produced per year, which gives you a fairly reasonable rate of return on your energy and greenhouse space that you're committing to growing mulberry trees. For another view, this is a field of mulberry shoots that have grown to their harvest maturity. And thanks to mulberry being a very fast growing tree, you're actually able to do this on a 1 to 2 year cycle. Okay, but now to jump over to some of the uses of silk in a Martian colony. So we're going to cover some of the domestic uses of silk first, but I want to pause on the slide for a moment to show you a great example of the harvesting process once silkworms have formed their cocoons. So by simply soaking the cocoons in a hot soapy water, you're able to loosen up the threads and then pull them out as individual strands. So an entire silkworm cocoon is actually made up of one long fiber that they just continuously extrude from their silkworm gland, or from their silk gland. This is the big goal of the caterpillar phase of the silkworm, the larva that you will typically see portrayed, and is to grow and develop this silk gland in order to have enough biomaterial to produce the silk for their cocoon. That'll become a lot more important too when we get to the biomaterial side of things. So, but from the domestic side, soft goods are really what we're focused on here. So clothing is a non-trivial element of a Martian colony, and silk is a great way to really solve this need. You can lean on synthetic fabrics quite a bit, but the drive for natural fabrics will always be there, and the ability to produce silk while you're also producing other valuable resources for the agricultural components of the colony and other elements. It just really makes sense to look at this as a market as well for the silk. So as I mentioned, it takes quite a few cocoons to make certain clothing items, and it takes about 600 cocoons for a typical shirt, and this is for the thickness there. So I say consumer textiles as well as an area where silk will be heavily consumed, but this is a very broad category. And while it might only take something like 600 cocoons to make a shirt, silk shirts are generally quite thin, and sheets can be up to 4 times thicker and even more depending on the quality of sheets that you're looking at. So it really increases the amount of silk that you're talking about for each item. But it's also good to note that it really bleeds out into a lot of other areas. So the materials for domes themselves on the interior lining can have silk included in them. And I know a lot of groups have talked as well about how these different materials have a different effect on the quality of life within these interior spaces. And silk really presents a good opportunity to increase the textural feel of these environments and to up the appeal from a user standpoint. It also bleeds into the space side of things a bit as well, in that silk has been used as a material inside spacesuits before and could serve that purpose as well on Mars. A couple other domestic avenues that can be pursued with silk too are some of the more enhanced silks. And so these are things that have been demonstrated in labs that would help to improve the industrial side of silk's capacity. So the orb spider is known to spin some of the strongest silk in the world, but the problem is that it only tends to produce short threads and is not capable of producing super long threads that would be much more useful in the fabrics that we're looking to generate. But it has been shown, and there have been transgenic silkworms made to the end of producing the silk in long continuous threads with a reasonably similar material property set to orb spider silk. So the main difference here is that it has a slightly lower strength, but the overall toughness is about the same. And it also can be produced in much larger quantities using silkworms. And it's been noted before too that one of the main issues in producing spider silk is finding a suitable talent body to work with the spiders. And it tends to be a very difficult process in harvesting, raising, production. So silkworms are really a much more comfortable organism to work with on that front. Another novel way of using silk is producing graphene silk, and this actually doesn't involve transgenic silkworms. This simply involves getting a liquid suspension of graphene and applying or mixing it into the silkworm's food if you're using a powdered chow, or spraying it directly onto mulberry leaves. The silkworms then just ingest the graphene along with the mulberry, and it gets lodged in their silk gland and incorporated into the silk itself, producing a significantly stronger silk that's also conductive while retaining its biocompatibility. All right. And now we'll jump over to one of the bigger topics with the Mars Society of Philadelphia, and one of the much more fundamental uses of silkworms, and that's in the agricultural side of things. Just a second. Actually, I'm going to pause here before we go into agriculture to answer a question that we got in the chat, which is, are silkworms expected to produce more silk in 0G compared to on Earth? And so to that end, I have not seen any studies that have directly looked at this yet, though I know China was interested in landing silkworms on the moon to continue getting more data on them in a space context. They did not end up doing that with that lander, but it was proposed, and I'm sure they're still interested in that. But as to the probability of whether or not that would occur, and this is again without any studies really to back this up, I would guess that no, they would not be expected to produce significantly more silk unless you saw an enlarging of the overall silkworm. They're already producing quite a lot of silk in their silk gland. It actually comprises something like 40% of their overall body mass by the time they get to the cocooning stage. And really, with that sort of dynamic, you're not limited by gravity in any significant way. It's only if it were to lower the stresses on the organism enough overall that it had the extra metabolic energy to put into the glands. But more than anything, that might just lead to earlier cocooning potentially, or other different growth abnormalities. So it's hard to really say more silk necessarily, but I wouldn't guess so. But that does, well, so one additional thing to note here is that it's not just the silk that we're looking at with silkworms. So it's also the pupae that comes out of the cocoon when we remove the silk. So like the pupa of the Pokemon, it is a cocooned organism that is essentially in a dormant state and is in transition to the silk moth, but is really dormant in and of itself, and is killed during the silk harvesting process. So that organism, though, still contains all the nutritional aspects of the silkworms themselves, and it is consumed as a food in a lot of the world, and can be used as a protein meal in a lot of food substitutes and health products out on the market today. But I'm really proposing that we look at these silkworm pupae as a feeder organism to a lot of the other organisms that will be used in the Martian biosystem. So depending on which way you want to go with this, and I'm really looking at the silkworms purely as a protein and nutrition source, and we'll get into the context of that in a second. But from the aquaculture side, this would be being fed to fish, or in other contexts, it could be to chickens or other livestock as just a general protein substitute. But part of why I lean into the protein is that silkworm pupae are 55% protein by mass. So that is really a fairly high concentration, and insects are known for this as well. Of having a very high concentration of protein. So it really makes them a very significant source of this protein as a feeder for these fish. And particularly when you look at the quantity of cocoons that we're looking to produce, and thus the quantity of silkworm pupae, you can get something like 100 kilograms of protein per cubic meter per year. So that is a very good benchmark to place against other protein production sources like black fly larvae or some sort of bioreactor. But given the other minimally intensive constraints of silkworms, specifically just producing mulberry leaves or using mulberry powder, it is a very low intensity way to produce this protein, especially if you're looking at lowering personnel requirements or automating the system. Another great aspect of silkworm pupae as part of the food chain and the greater food web of Mars is that it helps to reduce disease transmission. So these higher trophic order organisms like fish, like chickens, require certain components in their diets, including protein and fats. And in a lot of cases, what is fed to them in their diet is fish meal in the case of fish, or bone meal derivatives for chickens as well, but things that are well bioprocessed or well processed and potentially sterilized, components that came directly from the same organism. So fish to fish as fish meal in their food, or bone meal for chickens, that could potentially lead towards pathogenic transmission, and it promotes the development of parasites, or promotes the cycling of parasites. And while there will be many layers that can be placed to screen for this sort of thing in the broader Martian agricultural system, using silkworms is another foolproof way of removing the fish meal or removing bone meal or other protein substitutes that are animal derived from the animal feed, and thus reducing the potential of parasite cycling or other species or intraspecies disease transmission. And one of the more base level agricultural benefits of silkworms is that a lot of the waste that is produced during the larval stages, specifically the silkworm poop and the leftover leaf and food remnants, are great compostable material. Silkworms themselves have been shown to have antimicrobial and antifungal properties, antioxidant as well. So it's something that could help improve the overall health of your active compost system. And they're providing just a large quantity of bulk material for that active composting. One thing to note too, for those interested in a bit more passive designs, is that the amount of compostable waste that we're talking about here, given the number of cubic meters that you're looking to produce, it really is. So actually, to jump back on that for just a second here. So the metric here is 1 cubic meter for about 250,000 larvae. So about 1 cubic meter per 2 cubic meters of silkworm growth area. And so when you're talking about that on a year-long scale for, say, tens or hundreds of meters worth of cubic meters worth of silkworm production, then you're really talking about a significant amount of heat that can be generated through that compost, which could then be used in a greenhouse scenario as another source of heating. Another great thing to look into a bit more deeply here, I don't quite have time today, is the benefit of bioavailable nutrients, both in the compost and in fish meal or fish food and other feed products, specifically because it's coming from an organism that is part of the food web that these creatures are used to. It helps in the uptake, and also the silkworms were an organism themselves and thus have a ton of micronutrients and other beneficial compounds that allow them to survive and can be passed on through the food system. But the last large area I want to talk about, which is one of the most technologically interesting, is bioproduction in silkworms. And so what we have here is a silkworm that has been that is a transgenic silkworm that has the GFP gene incorporated into it. So GFP is green fluorescent protein, and it's often used as a marker protein to show that the vector process has gone well, that you have successfully introduced the new gene into the target organism. And so a lot of studies have used that, but once you fully develop the organism, you can remove or modify those reporter genes in ways that make them more useful or less intrusive to the overall bioproduction process. But so for those of you who might not be as familiar with the bioproduction world, some of these numbers may seem quite small, but they are definitely useful and relevant in this context, and they will be able to provide significant support to the Martian community. And one note here too is that all the things I'm going to be discussing are at research level, so yields tend to increase as you shift towards a manufacturing focus. But in general, raw yields above 10 milligrams of protein per individual larva are generally possible. And this is something that when you scale up into the millions leads to a very significant amount of protein being produced. And so things that produce, we're talking something like a kilogram per meter cubed a year of protein production. So the nature, and typically protein is produced in the cocoon itself, in the silk fibers. So that's really the context I'm going to focus on. But the silkworms themselves, the pupae that is then harvested, for a transgenic silkworm, you can modify it to have beneficial properties in that organism as well, if it were to be maybe an enhanced antimicrobial aspect to improve whatever end use you're looking at. But specifically within the silk, there are two main components, the sericin and the fibroin. Both of these have multiple proteins making them up, and there are several vectors that have been identified to put genes into many parts of those different proteins. But the range of yields is quite large, so some proteins are still looking at the micrograms per larva range, and that then does scale up much more slowly. But to go over just some of the big areas that are some of the bigger protein production platforms that have been established, I really want to focus on some of the growth factors and therapeutic therapeutic compounds like PDGF, platelet-derived growth factor, which is used in a number of different biological contexts, along with monoclonal antibodies. So I found insulin as one of the proteins that have been produced in silkworms to be quite interesting. And I wanted to take a moment to talk about this GAD65 insulin fusion. So this is a specific fusion protein that GAD65 itself is designed to help stimulate the body systems at large and improve the response to insulin and response to glucose stimulation in patients who are type 1 diabetic. So this is different than the typical insulin usage to manage sugar and is considered more therapeutic towards the condition overall rather than just treating the sugar intake. Though it should be noted that insulin itself in a bioactive form has been produced in transgenic silkworms. The ability to produce collagen and human-like collagen in silkworms as well. And so specifically, this is recombinant human collagen, not just a similar derivative. Is very helpful for biomedical and biotechnological applications as well. This is a growth medium for a lot of very delicate cells. So pluripotent stem cells are typically grown on a matrix that helps them maintain their non-differentiating complex, and it has been shown that silkworm-derived proteins are capable of maintaining that undifferentiated states in induced pluripotent stem cells. And so some of the more common materials that tend to drift over into the bio side too are your basic medical needs. So we're talking sutures, bandages, and gauze, and this goes back to the biocompatibility of silk in its natural state. So some of the other more interesting things as well that you can get from simply processing the silk proteins into a more monomeric form are cosmetics and a lot of antioxidants that are just generally taken as health supplements now. And notably, some of the health supplements can be derived from the pupa as well, but it's also useful in more broad scale medical applications as a pharmaceutical carrier. That it tends to be inert and, again, biocompatible. Okay. And as we get to the end here, I want to take a moment to also talk about the broader impacts here, now that we've looked at some of the different areas that silk can be applied to using silkworms as a fundamental part of the Martian ecosystem. So, of course, it would be good to be able to produce a lot of our goods on Mars. And the greater diversity of goods that we're able to produce, the greater diversity of consumer wants and population needs that we'll be able to supply. So a lot of this too cuts out some of the questions that are a lot more pertinent when we look at a permanent civilization on Mars, you know, specifically spoilage time and the ability to generate fresh resources on demand. And it's things like that that will really set apart an independent Martian colony versus an outpost or a, you know, business-driven institute on Mars, which just may be constantly resupplied with things and not really be looking at in situ resource utilization. It specifically develops more complex resources as well. So we're not talking about just harvesting the raw chemicals that could be applied to a greenhouse situation or even in aquaculture. We're looking at complex proteins that will help to develop more robust organisms on Mars and allow them to thrive and really focus on the breadth of the Martian experience and not just the subsistence side of the nutrition, the agriculture, the medicine that will be associated with these silkworm products. There are market aspects to this as well. You know, the more we're able to produce, the more leverage and flexibility we have towards the other areas of the Martian economy. And that's really one thing that I think the autonomous nature of silkworms, their ability to grow just by being fed continuously, really opens up. And the end point of all of this is to help really inspire Martian culture. You know, silkworms might serve as an industrial organism. They may be the main source of our clothing and some of our pharmaceutical needs on Mars, but really they can be an emblematic and unique difference that sets Mars apart. You know, we are reliant on a much more circular ecosystem because of our isolation from the resources of Earth. And I think it's really important to look at that context when you're looking at how we're looking to structure our future biosystems. All right, and with a couple minutes left, I want to open it up to any more questions. And I've got some references here if anyone wanted to look into a few more of the uses of silkworms and their produced biomaterials in specifically the biotechnology context. Oh, and so I have neglected to put my email on here, but if anyone would like to get in contact with me to discuss this a bit further or share some resources on this, or if you're interested in hearing a bit more about the Mars Society of Philadelphia, I can be reached at Stellie, that's STELLIE, at marssocphilly.space. That's MARSSOCPHILLY.space. So silk as an additive. So the first question is, has silk been used as an additive to make concrete stronger? So I have not seen any research towards this. And one of the biggest issues with incorporating a lot of organic materials into concrete is that organic materials can tend to change over time, particularly if they have access to gases and liquids from the outside, which concrete is typically a bit porous. So that does tend to be an issue. Maybe less of an issue in the dry, arid conditions of Mars, but typically I have not seen a lot of fibrous concrete additives, and I have not seen any silk being added to concrete. And second question from Will F. Are the biopharmaceuticals being produced in the body of the pupae, or was there an ability to produce them via the silk gland? Great question, and sorry if I didn't make this clear. So it depends on where you're putting your gene. So in a transgenic silkworm, so assuming you take a gene for something that you want, say you're looking at insulin, you take your gene for insulin and you put it into part of the silkworm genome. typically they're looking to put that into part of the silk. So either the silk sericin, which is the outer coating of the silk, or the silk fibroin, which is the inner core of the silk. So you can put the gene into either, but wherever you put that gene, it's going to then be produced there. And so in either of those two, it's going to be part of the silkworm silk gland. And remember that silk gland is 40% of its overall biomass. So you're still getting a huge proportion of the energy devoted to that. Or you can put it into genes that are associated with the silkworm pupae itself. So you can do that to the pupae, but it all is going to depend on where you put the gene. And so to get a bit more into the biotech of that, depending on how you introduce your gene, what vector you use, how you structure the construct that you're putting into the genome of the silkworm, you can definitely have off-target effects here. This is a part of what makes a lot of the newer technologies to introduce new genes so novel, is that they are very specific in where they put genes. I've not personally looked into CRISPR in the context of silkworm, transgenic silkworm development, but I'm sure that people are looking into it, and those technologies are broadly applicable to this. All right, very good questions, guys.
Yeah, thanks, Stellie, for coming and giving this presentation.
Yeah, thanks for having me. It's been great. Looking forward to hearing more great talks throughout the conference.
Okay, and I guess we're going to have a half-an-hour break, and then there'll be the main evening plenary sessions.
Awesome. All right, see you guys later.